Hello everyone, welcome to the first hormone in the plant that been discovered, auxin. Was discovered by Charles and Francis Darwin, the same Darwin that went into a beagle board in order to came up with their idea about evolution. So Darwin, the father and the son, interested in the fact that the younger plants tend to bend toward the light. So his experiment was using coleoptile, which is like a cap of a pen, but inside the cap is actually the leaves or the shoot of the grass family. So they discover that if the coleoptile was given with the light from the just one direction, the plants will bend toward the light. But if the tip of the plant was cut off, the rest of the plant will not bend anywhere. And if the tip of the plant was covered with a metal cap, again it will not bend. And later, different scientists try to do more experiment about the coleoptile. So the first group, Boysen and Jensen, they inserted sheet that is not cannot be passed through by water insoluble substance. If they put the mica sheet in the shaded side, there will be no bending at all. But if they put the mica sheet on the illuminated side, the plant can still bend toward the light. And the next experiment is to remove the tip and put the gelatin sheet in the middle. And again, this gelatin sheet can be passed through by water soluble substance. That means the shoot tip can bend toward the light. From this, all the experiment can be summarized that at the shoot tip, the plants synthesize some growth stimulating substance which can be perceive the light and travel down to this part of the plants and cause more growth that allow the plant to bend toward the light. And that substance is actually water soluble. Later, scientists find out that this hormone is named they name the hormone auxin, and they isolate the hormone from the plants. And auxin was can be synthesized in the plant through two pathway. Let's take a look at the structure of the hormone. This one is indoacetic acid. Auxin is actually the broad name. There are so many molecules or chemicals that can be auxin, and indoacetic acid is one of the auxin hormone. It is the natural producing auxin. And if you look at the structure, the chemical structure, you can see that it's quite similar to tryptophan, which is one of the amino acid. And it can be synthesized through either tryptophan-dependent pathway or tryptophan-independent pathway. That means the first one requires tryptophan, the second one can bypass the tryptophan. The production of auxin is mainly occur in apical meristem, especially the shoot apical meristem, which is the major source of auxin production. The other part of the plants like buds, young leaves, and other active young part of the plant can produce auxin as well. Auxin will work only in some range of concentration, not too little and not too much. If there's too much of auxin, it actually has some negative effect to the plants. So auxin can promote increase the plasticity or it mean flexibility of the plants by using acid growth hypothesis, which I will talk about it in like 2 slides from now. Additional auxin function is to promote phototropism, gravitropism, as we will see in the experiment in lab 2, not lab 2, lab 8. The next one is polar transportation, which is mean auxin will can be transferred in just one direction. It's not just go everywhere. It has to have some direction from the top to the bottom only, which can be determined by protein transporter, which is like a gate in the membrane of the plants. The next one is vascular differentiation. That just mean um, auxin can help where the plant will build the vascular tissue. The next one, fruit development, producing fruit, embryogenesis, more like the first stage of the plants after fertilization to build a embryo of the plants. Formation of leaf primordia. Leaf primordia is just like small part of small leaves, just baby leaves like that. Formation of lateral root. Lateral root is just the root that produced from the other root. Apical dominant is just mean the top of the plants will inhibit the shoot from the lower branch of the plants to grow. Cell division it just means dividing more cells, and delay of leaf abscission. Leaf abscission is more like falling of the leaves, so auxin inhibit leaf falling. And as I mentioned earlier about the concentration of the auxin that will have the response to the plants, so in a very low concentration of auxin or IAA in this part, the plant will not quite respond to grow more or grow bigger or less. At some certain concentration, the root will start to respond in positive way. And this is lower concentration compared to the shoot does. And at higher concentration in the root, if the root has more auxin above beyond this point, the root will not respond to higher auxin in a more positive way. If you look closely at this position, at this concentration, the root will not respond very well. It means like auxin will inhibit cell elongation if you use the high concentration. But at the same concentration right here, the shoot will respond in the positive way. The concentration that inhibit cells in the root to enlarge, that concentration promote the cells in the shoot to elongate it. And if you put too much concentration, the shoot will start to get inhibited by high concentration of auxin as well. So this graph is quite important, and I like you guys to understand how it works in this graph, because you will need this idea to explain the bending of the root and bending of the shoot in response to gravity or gravitoprism. Alright, let's talk about acid growth hypothesis. I think I mentioned about this in the lab video already, but it's better, right, to give you more explanation again. So, this is how auxin can govern or regulate how plant cells can get bigger or longer. This is just the before auxin action cells, and this is the after picture. you can see that they grow longer or higher. If you focus on this part of the plants, the left side is cytoplasm, the right one is cell wall. The left one cytoplasm, the right one cell wall. In the middle is the plasma membrane. And here you have a proton pump, which is like a gate that allow proton to travel from cytoplasm to the cell wall. And before having auxin, you can see that here is the cellulose microfibril. They have some bond connecting each fibrous each fiber together. So the cell wall is packed and stiff. They cannot be bending or expand very much. What auxin does? Auxin will stimulate the cell wall expansion or loosening of cellulose microfibril. How it does? It acts through acidity of the cell wall. Auxin stimulate the proton pump to pump more proton into the cell wall, and expansin, an enzyme that loosening the cell wall or cellulose microfibril, they love proton. So auxin give what expansin like, and expansin will expand or loosening the cellulose microfibril. Once the cell wall is loosening, the cell will have a chance to elongate it, but the cell will not elongate it if there is no other pressure to push the cell wall from inside toward the outside. So that pressure is the turgor pressure from water. Water will get into the cells throughout the water potential. Inside the cell usually have more solutes such as sugar, salt, for example. So the water will rush into the cells and give the turgor pressure, and the plant cell will expand toward the other side. Alright, this is how auxin promote the cell elongation. The next function of auxin is to promote formation of lateral and adventitious root formation. So lateral roots mean root that emerge from the older root. Adventitious root is the root that form from stem or leaves. So auxin can promote the formation of both lateral and adventitious root. For the lateral root, in this picture I'm showing you the action of auxin to promote cell division in the pericycle, which is next this tissue. The pericycle is the outer layer of vascular tissue right here. So auxin promote cell division right here, and the lateral root will grow out. But in the case of adventitious root, I explained this already in the last video. Instead of producing more cell division in the pericycle, the stem has cambium tissue, and auxin promote cambium to divide more, and you get the adventitious root in the stem. The next hormone that we will talk about is gibberellin. Gibberellin was discovered in Japan in the year 1926 by Mr. Kurosawa. He discovered the right disease called Bekane. That just means foolish seedling. The meaning or the symptom of this disease is the rice will grow very tall but very fragile. They will weaken and collapse and then die. This disease was caused by infection with fungus Gibberella fujikuroi. This is the old name, but from the old name you can see that the name gibberellin was come after Gibberella, and the extract of the infected plants contained the hormone gibberellin, and later it was crystallized and named gibberellin. The chemical structure was elucidated, and like auxin, gibberellins is just the common name or the name that many chemicals can be named gibberellin based on their similar structure and the activity. Nowadays, there are more than 110 known gibberellins, and most of them are inactive. And for the function, GA can in short, I will call it just GA. GA move through xylem and phloem without direction, without polarity as the auxin does. Because I told you earlier that auxin they they will move at one direction at a time, but gibberellin can grow wherever the flow of xylem and phloem goes. All right. So what are function of act of GA? GA can promote similar processes as the auxin does, just increase the stem growth, and is involved in nearly all of the same regulatory processes in plant development. GA promote other process as well. Like in this picture, you can see that these two cabbage they do not receive more gibberellin, but the other guys, these three one, you can see that they produce much longer stem and also promoting flowering. So GA can promote flowering in release bud and seed dormancy. This just means in general the seed will decide whether to germinate or not by having the two hormone in balance. If the seed receive more gibberellin or receive more GA, it will go toward the germination tendency. For the seed that has too much or more of abscisic acid, it will go into dormancy. That means it will not germinate. So the next function is to induce seed germination in plants that require stratification. Stratification is the process that the plants require a cold treatment before germination. Without cold treatment, these plants can induce seed germination with an action of GA. So in industry, application of GA, especially GA3 on the grapevines can produce longer stem, and the plants will produce bigger and more grapes. Increase in the yield of sugarcane. You can see that internode of sugarcane will be elongated. Hop is the earth that used for beer production. And the next one is cerali. If you go to the grocery store and if you like to eat cerali as a snack, you can see that the part that you eat is petiole of cerali. The next one is to delay aging of orange skin. The next one increase seed production in conifer. Can have you ever eaten like pine nuts, which is one of the nut that is very expensive? It's produced from the pine tree or conifer. The next one is induce the starch digestion in the mouth. that used for beer production or brewery. You you if you recall the alpha amylase experiment, um, alpha amylase can be induced by application of GA to release alpha amylase, and amylase will digest starch. That process is also occur in malt in the brewery industry. The next hormone is cytokinin. Cytokinin is promoting cell division, and in 1913, scientists named I will not attempt to pronounce his name. Anyway, he discovered that the wounded potato have more cell division when he applied phloem exudate from various plants. Phloem exudate is just like phloem sap from other plants. The plant cell of the wounded potato will divide it faster. And later, another scientist, Overbeek, in 1941, discovered that coconut water contains substance that increase growth rate of tissue and embryos. 9 years later, Mark Carlos Miller identified kinesin, which from herring sperm, and together the herring sperm containing kinesin, and he put auxin as well. These two hormones promote cell division of parenchyma tissue of tobacco plants in tissue culture. In this picture is the various combination of the concentration of kinetin, and auxin. No, none of the hormone will have this small callus. And callus is just the mass of the differentiated cells that will not decide whether to grow to some specific cells. So if you keep kinesin at zero concentration, zero milligram per liter, and keep increasing auxin, you will see that at some certain concentration, um, the callus will develop more root. Similarly, if you keep auxin at zero concentration and keep increasing kinesin, you will get more shoot from the plants. And if you give both in the similar or at some certain ratio, you will get cells that very big but still not decide whether to be a shoot or a root, for example. Here is the structure of cytokinin. So this is kinetin, and this is transiatin, which is the naturally occur cytokinin in the plants. The structure is quite similar, and actually kinetin can be synthesized by a human as well. Alright, function of cytokinin. It promotes cell division in shoot and root, increase leaf senescence. Leaf senescence is mean aging of the leaf. Apical dominance. It has some counter effect of auxin. Auxin will inhibit the growth of the lower bud, but cytokinin will promote the growth of the lateral bud. Cytokinin will also promote formation and activation of apical meristem, gametophytic development, or development of gametes. That means like producing eggs and producing sperm. Promotes promotion of zinc activity. It just mean zinc and sauce. Sauce is wherever the sugar is produced is more like the leaves can do photosynthesis, and the products of photosynthesis is sugar. It is the sauce that um, the sugar can travel from the sauce to the sink tissue where it cannot do photosynthesis, cannot make their own food. The next function of cytokinin is about vascular development, similar to auxin, breaking of bud dormancy. As I have mentioned about apical dominance as well, and increase of uh, interaction of plants with biotic and abiotic stresses. Stresses caused by something that not involved in living organism, more like light stress, too much light, too little light, too much water, too little water, high temperature or cold temperature, for example. For biotic stresses, the stress that caused by other living organism like fungal infection, virus or bacteria infection from pathogen, or herbivory got eaten by an insect, for example. Um, it's also involved in symbiotic nitrogen-fixing bacteria or avascular mycorrhizal fungi, pathogenic bacteria, fungi, nematode, and viruses, salinities and drought stresses, and macronutrient. And apart from plants, some certain kinds of bacteria can produce cytokinin as well. So in plant biotechnology or molecular biology, we exploit the fact that this bacteria can transfer its genes into the plants. What it does because the plants, not the plants, the bacteria named Agrobacterium tumefaciens, they have some piece of DNA that can transfer to the plants, and in general, this bacteria it contains the genes that can produce cytokinin. The gene that encodes cytokinin production will be infected to the plants, and plants will have the symptom that um, cause more like cancer cells. It produce more cells like because cytokinin promotes cell division and produce a lot of cell mass. We call it crown gall disease. But what the scientists exploit is they use the other part of DNA that has the ability to move the other whatever DNA the scientists want instead of the cytokinin synthesis gene, and this part of the DNA can be transferred to the plant. So scientists can put the gene they like in replace of cytokinin production and transform it to the plant cells. If they remove the cytokinin production synthesizing genes, the plant will not get the disease. Alright. The next hormone is abscisic acid. Abscisic acid is the so from the beginning to right now, we learn about three hormones: auxin, GA, and cytokinin. These three hormones more like promoting growth. The cell get longer, grow bigger. The GA can promote internal elongation, and cytokinin promotes cell division. But from now, abscisic acid and ethylene, these two hormones is more like inhibiting growth or promoting aging. Abscisic acid is the substance that can inhibit the, the growth of the plants. In 1949, a person named Thomson Hamsberg shows that substances he named dormin at that time. The dormin is just came from the word dormans. Dormins produced in dormant buds block the effect of auxin. In 1963, three independent groups discovered a growth-inhibiting hormone named abscisic acid, or ABA in short. Slug later tested to be the same thing as dormin, and ABA is synthesized in chloroplasts or in plastic from carotenoid pathway, and it can be found in fleshy fruit to prevent seed germination. As I mentioned about seed dormancy and seed germination in the GA part, I mentioned that GA will promote seed germination, but abscisic acid or ABA will prevent seed germination and promote seed dormancy. APA also function in delay um, seed germination and response to drought stress, mean too little water. This is the structure of ABA, which is quite similar to carotenoid. In carotenoid, you have more of this structure. This chain will get very long, and you have the ring as well. Here is the corn on the cobs that you can see that some corn kernel will start to germinate. This is because this corn kernel was um, the results of the plants that cannot produce ABA or ABA insensitivity. They will germinate before they are ready. Let's put it that way. And ABA is also controlling uh, stomatal posture. Here is the stomata right here, and besides the stomata, you have gut cells. These two gut cells will regulate whether it will open the stoma or it will close. And the opening of stomata is governed by blue light, and closing of stomata is governed by abscisic acid. Here how it works. So you can notice that here in the opening of stomata, the gut cell is very plump, very swollen, but in the closing of stomata, the gut cell is shrink and the stomata is closed. So the idea is about water potential in the plants. If you want the gut cell to get swollen, you need to have more water inside. More water inside mean more solute in the cells. Here you have cytosol in the yellow part and extracellular or outside of the cells in the blue part. Inside you need to have more salt and more sugar. So what blue light does will increase the concentration of both sugar, sucrose, and increase salt accumulation. And when you have the two solutes, you have more water to get into the cells and promote swelling of the cells, and the cells will open the stomata in the middle. So for the detail, blue light can be perceived by the molecules called phototropin, and this is the protein. These proteins will change the structure once it receives the blue light, and what it does next is to stimulate the proton pump to take out the proton, and it will stimulate differences in charge particle between outside and inside. Outside you have more positive charge, and once you have more positive charge, inside you have less. So the cells try to keep the balance by importing potassium ions, which is the positively charged molecules inside the cells. But the potassium ions can be it can be neutralized with anions like calcium or nitrate from outside. So anion is stimulated, potassium inside, followed by calcium, not calcium chloride, sorry, chloride and nitrate. And they can form potassium chloride and potassium nitrate. And these two molecules act as salt. Again, blue light can stimulate starch degradation. When the starch degrades, you will get more sucrose, and both sucrose and salt can increase the turgor pressure, can lower the solute potential, and water can move into the cells and increase the turgor pressure, and cells will get swollen. This is how our plants open their stomata. The process of stomatal closing by ADA is doing everything in reverse of opening. So closings mean inside the cells they need to get rid of all of their solute. So ADA will promote the calcium ion movement to get into the cells. Abscisic acid will increase the internal calcium storage to release more calcium ions, and calcium ion will inhibit the proton pump. It will inhibit the potassium pump. It will promote the anion efflux, or it will get rid of all of the anions. So everything in reverse and increase the pH. So that means inside the cell you will not have proton, you will not have potassium, you will not have chloride or nitrate. So inside the cell, the water move out because outside you have more of the salt, more of the sugar, and then the cell get shrink and the stomata is closed. All right. So we're done with the four hormones. The next one is about ethylene. Ethylene was discovered firstly in Russia. At that time, many cities in Europe they do not have electricity, so but they have lighting by using lamps, and lamps was working from the gas, and one of the gas was actually ethylene. But um, ethylene was discovered to be a plant hormone by the fact that one scientist, he was very young, 17 years old. He working in the lab. The lab has a gas lamp, and one day he found that the plants growing in the lab was has something different. They have thickened stem and grow horizontally compared to the plants that grow outside of the lab. This is because ethylene from the gas lamp was leaked and produced abnormal growth of the seedling. So later, another person found that fruit can produce ethylene naturally and can be produced by flower, seeds, leaf, and root as well. Production of ethylene is vary under different concentrate conditions, like application of auxin, germinating seed that touch the soil can produce ethylene in order to form apical hooks. Can you, if you notice some of the pea plants, the the young leaf will bend more like a hook, and that's promoted by hormone ethylene. And moreover, ethylene promote um, responds to mechanical stresses like hitting some rocks or getting bumped into with an animal. Plant respond to ethylene by increasing fiber and parenchyma tissues that support the plants, inhibit cell elongation, and so you will resulting in shorter and sturdier plants, and promote leaf falling, promote fruit ripening. Ethylene can also trigger its own production as well. This is the hormone ethylene. The structure is very simple. This picture is if you put the plants in the presence of ethylene, the leaf will abscise and fallen, and the fruit will get ripened. Talking about fruit ripening, I like to show you this graph. It's about application of ethylene in some type of fruit. It will promote fruit ripening, but in some kind of fruit it's not having any effect because there are two kinds of fruit, climacteric and non-climacteric fruits. Climacteric fruit is more like apples, pear, banana, avocado, durian, for example. This fruit, during the ripening, they will increase respiration rate and increase ethylene production, and it's require ethylene to ripen. And this is the growth. For the non-climacteric, look at the broken lines. Non-climacteric fruits, the growth is same thing as the climacteric, but the respiration rate is not increased during the ripening, and the amount of hormone ethylene is still flat. So the fruit industry can control the ripening of the fruit like bananas by removing if they can store fruit in to sell it out of the season, they can store in the cool place that reduce temperature and remove oxygen. So the fruit will not have more of the respiration, and the plants will not produce much of ethylene. Once they they are ready to use that stock fruit for selling, they will put ethylene gas so the fruit will get ripened. This is easily can be applied to banana, for example. Ethylene can be used in promoting flowering in pineapple as well, and promote more female flower in pumpkin's family. I used to grow zucchini plants. It's the same family as pumpkins, and um, I have some problems as well because they produce a lot of male flowers, but not too much of their female flowers. So less female flowers, that means you will not get the fruits. All right. This is hormonal interaction. It's about apical dominance. Like I mentioned earlier, that the auxin is produced in the shoot of the plants, and it will travel toward the root direction. And along the way, it will stop growth of lateral bud. But if the top of the plant was cut off, there will be no more auxin, and cytokinin will promote the growth of the lateral bud right here. This is the real picture of the plants. The first one is the picture of the bud in the intact plants, and the rest are several days after the tip of the plant was cut off. Here, once the new tip, new leaf or new shoot was growing in size, they will produce auxin, and it will inhibit the lower lateral bud to grow. The next one is the interaction between cytokinin and ABA, auxin and gibberellin and cytokinin. All that we learned today is about senescence. Senescence just mean aging. You can see by the falling leaves during winter in seasonal the temperate zone in the planet. So this is like in September, October, early October, and just a week later. This is like 2 weeks apart. You can see that plants leaves can turn to orange and then falling, and this has about three phases. Initiation phase is the transition of nitrogen sink. That means the leaves will get a lot of nitrogen in order to make chlorophyll. The leaf just transition from being required from needing nitrogen to giving nitrogen, and photosynthesis decline and early signaling event. And then the degenerative phase is means the cellular constituents start to degrade. All the macromolecules like protein, fiber, carbohydrate, DNA, RNA start to degrade. And then finally, terminal phase, loss of cellular integrity, cell death, and leaf falling. Actually, the plants will recycle the nutrient, as I said earlier about the nitrogen sink to nitrogen source. Plant will recycle nitrogen from the older leaves to the newer leaves. And abscisic acid and ethylene promote the leaf senescence, but this effect was counteracted with auxin, gibberellin, and cytokinin. Other factors such as nitrogen deficiency, drought can speed up this process as well. Right. The last one, probably not quite last, about leaf abscission. Similar to leaf senescence, but leaf abscission is the process that the petiole from the leaf was broken apart from the stem of the plants. This in the region is called abscission zone. In this region was determined and produced long before the leaf was actually falling. Uh, and what happens is just like this part of the plant, the cells will has some cellulase, the enzyme that degrades cellulose, and when cellulose degraded, the leaf will get falling. There are three stages as well: stage 1, 2, and 3. The stage 1, high auxin from the leaf will reduce ethylene sensitivity of abscission zones. So auxin will inhibit the leaf abscission. The second one, abscission induction phase, a reduction of auxin, so no more auxin, or lower in amount of auxin from the leaf will increase the ethylene sensitivity in abscission zone, which trigger the abscission phase. Abscission phase is the synthesis of enzymes that hydrolyze or digest the cell wall polysaccharides, resulting in cell separation and leaf abscission. Timing of leaf abscission is regulated by interaction of ethylene and auxin, right? Like we have seen in this picture, ethylene is the primary regulator of abscission and cell separation between abscission zone, and auxin will suppress the ethylene effects. Alright, I think what I have in this video is about hormone. So we will see you again in the next video. It's about plants movement.